So we're gonna take what we started with, like how a muscle cell, a skeletal muscle cell generates its contractile force with that sliding filament theory. And we're gonna expand a bit, talk about how muscles, you know, because obviously that thing we saw was kind of an all or none. You like dumped calcium into the thing and all the actin and myosin were pulling. But we're gonna look at how we connect that up in the larger organs that are the muscles, like your biceps brachii or whatever. And then we're gonna also talk about different kinds of muscle fibers that are optimized for like sprinting versus, you know, long distance kind of running, things that are for, you know, different muscles for different kinds of jobs. Then we'll talk about the smooth muscle. And then we're gonna introduce the cardiovascular. And that will be kind of the end of our lecture part. And then for lab, we'll take a break. The lab is going to be the blood pressure lab. And I'll talk more about it when we start, but it's gonna have different segments. And again, we're talking right now about skeletal muscles. So let's first just review what we formally call excitation contraction coupling. I'm trying to excitation contraction coupling. So the basic idea, excitation is the signal coming down from some motor neuron. There's some action potential. Contraction is you actually have a muscle cell here. Again, it looks kind of striped because all of those sarcomeres, that alternating arrangement of actin and myosin and actin and myosin and actin and myosin. So here we're going to have our neuromuscular junction. Again, so the first step of this is there is a motor neuron in your CNS. Remember we talked about there's just one hop from somatic, in the somatic motor pathways, there's one neuron whose cell body is in the CNS, sends an action potential. This could be, again, like 3 feet long, this axon. Might be going from your spinal cord all the way down to your foot. And then at the neuromuscular junction, what happens? What is released at the neuromuscular junction in order to excite the neuron?
Acetylcholine to the nicotinic.
Exactly.
Cholinergic.
So the motor neuron releases acetylcholine, you know, which then is going to bind nicotinic cholinergic receptors on the muscle cell. What happens next? Oh, that's probably too small. Can you read that, or is it kind of a little small? What's going to happen next? Once the nicotinic cholinergic receptors on the muscle cell are bound by or binding the acetylcholine.
Does the action potential travel around?
Yeah, so that's going to open up sodium channels in the membrane of the muscle cell. And we talked about how the sarcolemma, the cell membrane of the muscle cell, is all full of those voltage-gated sodium and potassium channels that we saw in the axon of a neuron. So if you have sodium coming in on the membrane, that's going to depolarize that part of the membrane, bring it up above threshold, above minus 55 millivolts, which is then going to trigger an action potential across the entire sarcolemma. So you can actually measure these voltage changes on the skin. It's called an electromyogram. if you put electrodes on your skin and you start moving around, you can actually see voltages going up and down, because every time you activate and contract the muscle, there's this big voltage change across the surface of the muscle cell. So it's actually used in, clinically, it's used in research. I don't want to, maybe I'll put it in like EMG, electromyogram. Just like, the ECG, electrocardiogram, is electrical activity you measure on your skin that is due to your heart depolarizing. Electroencephalogram is the electrical signal on your skin that's due to the action potentials happening in your brain neurons. So electromyogram is just the electrical signals on your skin due to all of the muscle cells depolarizing during this process. So what happens next? We have electrical excitation spreads across the entire cell, and that triggers what inside the cell?
Calcium to be released.
Exactly. Calcium is released from the sarcoplasmic reticulum. And what does that calcium do once it's loose in the cell? So you gotta, do y'all remember? Kind of went in, it bound a particular.
It binds to the troponin.
Exactly. So that calcium maybe I should say calcium. Calcium binds troponin, in the sarcomeres there. And that troponin was that thing that is anchored both to the actin thin filament as well as to the tropomyosin, the protein that is blocking the binding site on the actin. So when the troponin binds calcium, it has a conformational change. It actually changes its shape. So this is going to move the tropomyosin out of the way and exposes binding sites on actin. Again, actin is the thin filament in the sarcomere.
And you said the tropomyosin is bind to both the actin and the sarcomere?
Tropomyosin is kind of in the little groove blocking the binding sites on the actin, and the troponin is grabbing onto both of them. So it can shift, as it changes its shape, it moves the tropomyosin up out of the way. Or if it's not bound to calcium, then it lets the tropomyosin sit and block the binding site. That makes sense. And then once the binding sites, what is going to bind onto the actin?
Myosin?
Exactly. So then the myosin heads bind to the actin. And that was the trigger, if you remember, the myosin binding the actin, that is going to create the power stroke. So the power stroke is when that myosin head grabs onto the actin and then snaps and pulls. And that's going to mean, the Z lines are going to get pulled closer together. So that's the actual kind of contraction that's happening here, right? So we started with the excitation, some signal coming down the nerve, but now we have contraction. We have the Z lines getting pulled together with force generated by the thick filaments, the myosin heads snapping in. Just to complete this, what happens next?
Does it have to do with the ATP?
It does. So what does the ATP do?
Once it rebinds, then doesn't it all go back to its original?
So the myosin, binds ATP, which allows it to detach from the actin and to recock. Recock. So, and then it's kind of ready for the next round. So you should understand all of these steps. These are all the steps involved in both exciting a muscle cell as well as what's going on inside the muscle cell that actually generates the contraction. And all of the muscle cells, all the skeletal muscle cells have the same basic story here. We're going to talk about some different varieties of muscle cells, like the fast twitch and the slow twitch. So they're going to be different in how they generate their ATP, or like how much actin and myosin they got going on inside, or maybe the specific versions of myosin they have that snap faster or slower. But if it's a skeletal muscle cell, this is the basic story in there. So you should make sure that this makes sense. Are there any questions about kind of going, and this is kind of what was in that. I actually made a link to the that little video that looked at this as well. So you can look at that. It's kind of pretty with kind of graphics and all that. Okie dokie. So now we can go, in fact, maybe I'm realizing after all this, I have, it sure would, images, muscular system. I'm going to just share the picture of this just so you can see it as a prettier picture. So again, this is this idea that if you're looking in this is like an let me make a darker one here. If you're looking at an overall muscle, like this could be your biceps brachii or whatever, and that is made up of all these bundled fibers. Like this here is an individual muscle cell. So there's going to be thousands and thousands of muscle cells all bound together. In anatomy, we talk about the different levels. There's the endomysium and the fascicles with the perimysium and the whole thing wrapped in the epimysium. But for our class, you don't got to know any of that. You just need to know a whole bunch of muscle fibers all bundled together to make the overall muscle. And then if you look at a single muscle fiber, and again, fiber is a synonym for cell. It's a long, thin cell, so they also call it a fiber. Same thing with nerves, right? Nerve fiber means a nerve cell or a neuron. Inside, we had those little organelles, the myofibrils, and the myofibrils were made up of all the actin and myosin, again, just alternating thick filament, myosin, and then more of it actin, and then myosin and actin. That's why it looks striped. That's why it's striated. And then if you look at the actual sarcomere, again, you can see thick filament, the myosin, which is sitting in this pocket of actin. So if you take this, if this little dude grabs and pulls, it's going to bring the two sides, the Z line, in closer together. It's going to burrow deeper into the pocket. But again, the length of the filaments are not changing. That's why they call it sliding filament theory instead of like contracting filament theory or something. So just, to give you a better picture there, that's kind of nice. Under the microscope, it looks kind of like this. That's why it looks striated. And again, don't worry. All these things people always talk about, H and A and I and M. That is back before we knew about actin and myosin. It was anisotropic, isotropic. Helle means bright in German. It was just words or ways to kind of talk about what they could observe, but they didn't actually know. For our purposes, all I care is that you know about actin and myosin pulling on each other. Again, myosin, a single myosin is got quaternary structure, these different subunits, and they have a tail and a head, and the head can be either cocked or it can spring when it binds onto the actin. La, la, la. This is kind of showing just actin, even though it's a thin filament, it's actually made out of tiny little subunits. This is kind of showing the tropomyosin blocking the binding sites when you do not have the calcium around. The troponin, somebody was asking about the troponin here is attached to both actin and myosin, actin and tropomyosin. So when the calcium comes in, see if this is kind of showing that. When the calcium comes in, the tropomyosin moves out of the way as the troponin changes shape, and then the myosin head can grab on. So yeah, you should go back and look at all your book and the online resources and all that. I'll explain this pretty nice. So let's continue. I'm going to go back to my whiteboard. So first thing we're going to talk about now is the idea of, let's look at, I want black. So first let's look at a single muscle cell. And we're going to look and think about the force it generates as it contracts. And this is just going to be kind of excite the motor neuron. So you send a message down that motor neuron, excitation, contraction, coupling, right? So you send an action potential here, then it's going to create an action potential on the sarcolemma, which releases calcium. The calcium binds the troponin moves the tropomyosin, the myosin head grabs onto the actin, and it pulls. And oh my God, we just generated a little bit of force. It was like a delay because it takes some time for all that process to happen. Normally, the way if you want to kind of like turn on a muscle cell, and this is kind of all or none, you can't say, this is called a twitch, if this happens. And typically what you do is you just kind of keep firing off messages down the motor neuron, and you basically just kind of fill up the thing with calcium. As that happens, the force comes up. They call it treppe. Again, a lot of German because the original anatomists that we're describing and physiologists were German. So treppe, little steps going up. But then you get some maximum sustained contraction here. And that is called tetanus. Not tetanus. So it's not the disease. It's not like lockjaw you're getting from botulism or something. Tetanus just means you are activating this little motor neuron that's talking to the muscle cell, and you're getting this maximum contraction. Again. But then it starts dropping off. So even if you just keep activating the muscle cell, you start losing contractile force, and this is called fatigue. Exactly why muscle fatigues. Oh, hold on, I usually shut my window. It's obviously my complexion will probably look less wonky if I shut my blind there. This is part of like when I first started like learning about, you know, science. I kind of just figured you keep going into a deeper and deeper, like more detailed textbook and you find the answer. It's like no matter what you want to know, you know, you just have to go to the more advanced texts or something and figure out what you want to know. And one of the things that is really kind of cool about science, hopefully you're kind of going to understand more and more as we go through this class, is we hit the limit pretty quick on what we know. Even something as simple or as fundamental as why does a muscle cell stop generating force even though you keep exciting it? We don't totally know. And in fact, the story has changed. When I first started teaching this class, they had a pretty good theory about the buildup of lactic acid and was affecting things in a way that was minimizing the ability to generate force. And then later research was showing like, no, that actually isn't true. There's something else going on. Nobody, at least to the last that I actually was following the literature, this was still a bit of a mystery. Basic sliding filament theory, the fact that actin and myosin are pulling on each other and sarcomeres are shortening, that we've felt pretty good about and has been kind of continually reaffirmed since the 50s. But there's a lot of things about muscle physiology, something as fundamental as this even, that we're still trying to figure out. So just kind of giving you a sense of, while we know a lot, you know, the rabbit hole goes so freaking deep. And even some very basic stuff becomes kind of mysterious when you're trying to understand it.
You said fatigue is when your body just stops generating force?
The muscle cell.
Right. Okay.
So there's another concept of like central fatigue, central nervous system, like, oh my God, I'm so tired. Like, you know, at some level, it's hard for me to get up and pick up my stylus and draw because I just want to take a nap. But that's something else. That's happening for my brain. This is something physical with the muscle cell. You keep activating it to contract, and yet it stops generating force. So it's something physical in the muscle cell rather than something in your brain where you're just tired and don't want to move. So yeah, central fatigue is this other concept. And people talk about central fatigue.
And we don't know why this type of fatigue happens?
At least last I looked, we still weren't sure exactly. So this is versus, you know, central fatigue is kind of just being tired. So this leads to a problem though. In fact, we'll come back to this. Well, so there's a couple of issues here that we're going to have, this is going to lead to. One is a single muscle cell, at least a skeletal muscle cell. If you kind of start, you know, sending messages down the motor neuron, it's going to have a certain level of contractile force, you know, before it fatigues even. It's kind of all or none. You either, you know, it has a certain number of actin and myosin all pulling together as long as you're flooding it with calcium. And so you don't really have the ability to have a skeletal muscle cell, like pull more strongly or pull less strongly. We're going to see that's different. When you get to cardiac muscle and stuff, you actually can have graded contractile force due to different processes that are going on. But for the skeletal muscle cell, it's either contracting or it's not contracting. And so that's going to lead us to have to get creative to how do we actually, you know, I actually don't have my cat here. Hilarious. But let's see, I can use my mascot. This is my like coronavirus coyote here. Oh, coronavirus. Oh, I hit it. You can pet things. You can whack things, right? You can generate a really hard force. You can generate really tender, gentle forces with the same muscles. How the heck did he do that? How can the same muscle do a hard force or a gentle force, even though it's all or none when you think about an individual muscle fiber? So let's take a look. Interesting question, coronavirus coyote. It's going to come down to the idea of motor units. Now, we're not getting punchy here, don't worry. Motor units. Let's see. So motor units, the idea of a motor unit. A motor unit is, by definition, I'll make more sense when I draw the picture. It's going to be a motor neuron. In fact, let me just use the typer. Motor neuron and the associated muscle cells that it innervates. So that's the basic idea of a motor unit. And so let's look at an overall muscle. If you think about, let's say this is your biceps brachii muscle, connected to your bones and whatever. So this is the overall muscle. So this is the muscle as an organ, made up of lots of muscle cells all bound together. So I'm going to have, let's do our red motor unit. So here's some motor neuron, and it's going to be innervating. I'm going to just draw a bunch of red muscle fibers. So it's going to activate my red muscle fibers. I have my blue motor unit, which is going to innervate, connect up to my blue muscle fibers. I'll have my green motor unit. It's getting messy. It's going to activate my green muscle fibers. So the basic idea here is if you have a muscle, you can, even though any particular muscle cell is either going to be pulling or not pulling, depending on how many motor units that, in fact, the fancy word, the term people use is recruitment. If you recruit more motor units, you're going to ultimately have a greater overall force generated by the whole muscle, right? So if I want a very delicate force, I only activate a few motor units. If I want a really strong force, I'm going to activate lots of motor units all at once. So even though any one muscle cell is either basically on or off, by recruiting more or less motor units, you can have a much stronger or more gentle force generated by the muscle as a whole. How many cells per motor unit? Do you all think?
1.
So if you think it's a single muscle cell does not generate very much force at all. So in general, you're not going to need to have such fine control to add or subtract the force by a single muscle cell.
Does it depend on the size of the muscle?
Yes, it's actually going to depend. I would say in general, like on average, let me give myself some room in here. On average, let me get out of caps lock. Maybe 50 to, a couple of hundred muscle cells per motor unit, meaning that you can add or subtract force, in amounts of, adding or subtracting 50 muscle cells, like something like your biceps brachii or something. It's going to be around like this. However, other muscles are going to be different. Someone was saying 1. I'm not sure if it ever gets down to 1, but it does get down to like, maybe, 3 or 4 or 5, can be as low as, 3 to 5. And what kind of muscles are going to need incredibly fine control of just adding a little more force, a little less force, really fine gradations?
Fingers.
Exactly. Like in your fingers, fringes. What the heck? In your fingers, your extrinsic eye muscles. So places where you have really fine control, you might have only a few. Other places, like that gastrocnemius, like your calf muscle, could be 500. Your calf muscle, thing that puts you up on your toes or not, that doesn't need really fine control. And there you might be adding or subtracting by 500 motor fibers at once when you activate or recruit or unrecruit the motor units. So that makes sense. The other thing we can bring up in here is that fatigue. When you turn on a muscle cell, it is going to fatigue eventually. So I can take my stylish little, like, here, my little Choco Cat tape dispenser, and I'm holding it up in a cool pose. And I could probably just stand like this for the next half an hour and just do my cool little Choco Cat tape dispenser pose. But the muscles that are holding my arm up that's made out of fibers that are going to fatigue if you keep activating them. So how is it that I can keep this pose going even though the individual muscle cells in my muscles are going through fatigue? Anybody?
Is it because a lot of cells are being activated?
Any particular cell that gets activated, though, is going to eventually start fatiguing.
They cycle through?
They cycle through, exactly. So what happens is you get this process. Let me give myself some room here. You get this process called asynchronous recruitment. Which all that means is, maybe this motor unit is going. Well, that highlight does not work well. Maybe this motor unit is going, and then these neurons are fatiguing. So this one goes off and takes a little break, and then this one takes over. And then this one is fatiguing, and then this one takes over. And then meanwhile, this one has recovered, so this is fatiguing. So as long as you have enough motor units waiting in reserve, they can just cycle. And a motor unit that is still fresh can take over while a motor, the neurons that have fatigued can recover. So that makes sense? So that's obviously, if you try to lift something really heavy, eventually you can't. What happens if I lift my entire chair up? I'm going to try to hold this, and eventually I have to put it down. Like, how come I can't kind of strike my pose and hold something heavy and just hold that forever?
You run out of ATP?
But more, think about what we were just talking about with the asynchronous recruitment. It's not really so much about ATP. It's about the fatigue.
You're using all the motor units that you have, so you're not able to cycle through them.
Exactly. If I am turning on most of my motor units to generate that really strong force I need, then there's not going to be any motor units waiting in the wings to like take over when the motor units start, or when the neurons fatigue, not the neurons, the muscle cells fatigue. So asynchronous recruitment only works if you're not like totally firing off everything at once, making a really strong force. If you try to like really have the muscle go as hard as it can, you're going to have to stop pulling and let the whole thing recover.
How many motor units do we have per muscle?
It really depends on the muscle. So it really depends on the muscle. Again, how many motor units, how many cells per motor unit, it really depends on which muscle you're looking at and what kind of function it's doing and what kind of control it needs, what overall force it needs to generate in the big picture. So yeah, if you remember like anatomy, there's like 614 muscles and they're all really different from each other. And there's other things we could talk about, how the fibers are connected either in parallel or at angles. You know, there's lots of cool stuff we are not going to talk about because I want to make sure that we get through all the material. But this is kind of essential. We need to kind of understand motor units.
So if a muscle's cramping from fatigue, what's happening there?
That seems to be more about the neuron. That is more the control of the muscle, where this keeps sending messages to the muscle to keep contracting. So my best understanding, at least, of cramping is it's not about the muscle tissue itself. It's about the neurons that are exciting the muscles.
Okay.
All right. So the other thing we're going to talk about for skeletal muscle is fast versus slow twitch fibers. But before we talk about that, we need to talk about how do muscle cells make ATP? Right? Because if you don't got ATP, none of this is going to work. So ATP, power source of all of this. And what's the classic way to make ATP?
Cellular respiration.
Yeah, totally. So one possibility is just to do cellular respiration. That's the classic. It's really super efficient in terms of getting a lot of ATP from every single glucose, but it's really convoluted and takes so many steps, right? You go through the 10 steps of glycolysis and then bring the pyruvates into the mitochondrial matrix and turn it into acetylcholine, which feeds into the Krebs cycle, and you're making all the NADH, and then the NADH contributes electrons in the electron transport chain, and then that pumps protons into that intramembrane space, and then the protons come back through that ATP synthase, and finally, at the end of it all, you're making some ATP. So it's efficient in terms of ATP per glucose, but it takes a freaking long time for that process to go through all those steps. And if you have a muscle with a lot of myosin heads that all want to unbind and resnap so they can go for the next round, you can't wait around. So there are two other ways of making ATP that make it way faster. So, you know, this is going to be aerobic, right? It needs oxygen for this to go. This is happening in the mitochondria. There are going to be a couple of anaerobic pathways. I did not, let me spell that better. So we're going to have two different anaerobic pathways. To make ATP. And both of them are going to be faster than waiting around for all the steps of cellular respiration of the mitochondria to happen. One of them you already know. What's just the first steps of cellular respiration?
Glycolysis.
Yeah, exactly, glycolysis. So glycolysis, if you remember, it only made 2 ATP for every glucose, but it happens pretty straightforward. It's just 10 steps and you got the ATP instead of all of the convoluted machinery in the mitochondrion. So even though you are only getting 1/16 as much ATP given the amount of glucose you're starting with, if you actually just look at how much ATP is showing up per second, it's actually way faster than cellular respiration. So if you are looking at ATP per second, this is actually two and a half times faster than cellular respiration. So again, it's not efficient. It's making only 2 ATP per glucose, but it's just if you really care about just right now, I want a lot of ATP fast, it is giving you ATP much faster. But you are going to build up lactic acid, which is going to actually have to get dealt with. Ultimately, it goes to the liver, and the liver converts it back into pyruvates and things and deals with it. So it makes ATP really fast, but it builds up this kind of waste product you need to deal with. Another anaerobic pathway that's even faster than this is what we call the phosphagen pathway. This is going to be one step. We're going to have this thing called creatine phosphate. Let me maybe. Creatine phosphate. So we have creatine phosphate, which is basically this molecule called creatine bound onto a phosphate. It's the same phosphate that's on ATP. So creatine phosphate plus ADP, which is again adenosine diphosphate. Remember, after ATP is hydrolyzed, you end up with an ADP plus a phosphate. So creatine phosphate and ADP, in one step, it just transfers the phosphate group. It's going to be the creatine, which needs to get regenerated, plus an ATP. One step. So this is going to be 4 times faster than waiting around for cellular respiration.
So when people take creatine, is that just allowing for more energy to be made? Because the ADP can attach.
Right. So it's an interesting thing. There are ambiguous results. People who've looked at creatine supplements, some of them say that it might help you, particularly with really just intense, impulsive kind of movements and strength. We're going to see this particular pathway is when you're really sprinting, when you really need the ATP really fast right now. So this is 4.5 times faster than cellular respiration. Glycolysis is 2.5 times faster than cellular respiration. Both of the glycolysis and the phosphagen are not sustainable. In glycolysis, you're building up lactic acid that you need to deal with. With phosphagen, you're breaking apart all the creatine phosphate, which has to get dealt with and regenerated. I'm going to draw this prettier in a second. What enzyme, just to kind of keep you on your toes, what kind of enzyme do you think is going to be responsible for transferring the phosphate from the creatine phosphate back to the ATP? Do you remember the naming scheme? enzymes that transferred phosphate groups. So we have creatine phosphate plus ADP, which is just an adenosine with 2 phosphates, becomes ATP, which is now adenosine with 3 phosphates, plus the creatine. So what kind of enzyme?
Something kinase.
Exactly. Awesome. So it's going to be a creatine kinase. As kinases, we've met them, we'll meet more of them. Enzymes, phosphorylating things, transferring phosphate groups is just a really common story. So if we look at those 3 ways of generating ATP, again, there was cellular respiration. There was glycolysis. There was the phosphagen system. Again, this is kind of the classic. This one is just how your cells are always making ATP. This can go on forever. Glycolysis, 2.5 times faster, but it runs out of the usually burning glycogen and builds up lots of lactic acid. This can only last about 1.5 minutes before it has to recover. Phosphagen system, again, 4 times faster. But this only lasts like maybe 10 seconds before it has to recover. So if you are really sprinting, you're going to be making lots of ATP using this. If, within 10 seconds, though, this is blown. You still got the ability to keep making ATP using glycolysis, but after this is going on, you need to recover. You need to get rid of the lactic acid and reconvert it back into other things that goes on in the liver. You got to deal with all the creatine that's been broken down. So you go into what's called oxygen debt. And this basically, the idea here is after using those anaerobic pathways, you need about an extra somewhere around 11 liters of oxygen on top of the normal oxygen you'd normally be breathing to recover. So, right, if you were, in fact, one of the things we're going to do today, and we're going to pretend we're going to do today, is like this step test where you're going up and down. Imagine you're just sprinting and running, and then you finally get back into your chair. You're sitting on your butt. You're not doing anything when you're gasping for breath there after you've just been sprinting. Your muscles don't need that oxygen when you're sitting there panting and recovering. You're in oxygen debt. So this idea of while you're actually sprinting, you're going through making ATP using anaerobic pathways. You're kind of borrowing, so to speak. You're getting lots of ATP right now, but you're building up all this lactic acid. You're breaking down all the creatine phosphate. And about 1.5 minutes in, you're spent and you need to recover. You need to get a lot of extra oxygen on top of your normal oxygen. And that's that kind of sitting there panting and kind of recovering from going into those anaerobic pathways. So now that we've kind of looked at these different types of pathways for making ATP, we're kind of ready to kind of finish our talking about muscles here and look at the fast versus slow twitch muscle. So, and again, I'm realizing it's better to use the typing tool here. Different types of skeletal muscle fibers. So even though all skeletal muscle fibers use sliding filament theory, have actin and myosin with tropomyosin and troponin and all this stuff, there are different kinds of muscle fibers optimized for different kinds of jobs. And I will kind of do a side-by-side comparison. One kind are called fast twitch. Fast twitch, AKA also known as, they can also be called glycolytic. And we're going to see that's because they are mainly using like kind of glycolysis and these anaerobic pathways, the other anaerobic pathways. They can also be called, I should say, AKA white. These are called white muscle or white muscle fibers. As opposed to the other kind, which are slow twitch. AKA, also they're called oxidative fibers, because these are the ones that are going to be using cellular respiration, using the oxygen, using pathways to make ATP. AKA red. This is red muscle. These are the ones, you know, we'll see it's because number of things. There's myoglobin, things to bind, hold oxygen in the tissue, way more vascularization in there. So we can talk about difference between these. Let's start with the functional differences, how they're different functionally. Functionally, they're different. Fast twitch contracts way faster. Let me use a smaller font here. Gosh darn it. I thought I was in it. Okay. Contracts quickly, fast. Whereas slow twitch, true to its name, contracts more slowly. Part of this is due to actually different subtypes of myosin. The myosin in fast twitch muscle has heads that snap faster. So part of this is actually just due to the mechanics of the snapping myosin head. Fast twitch also has much stronger contractions. It's going to be weaker contractions when you're looking at slow twitch. That is going to be due to the fact that fast twitch has more myofibrils, more of those organelles full of actin and myosin. If you look at a skeletal muscle cell, remember it was like a cell, and then it had all of those myofibrils inside. If you look at a fast twitch, it's got a larger diameter and way more myofibrils all pulling in parallel, so it's going to generate an overall bigger force. So these are going to have larger diameters. Gosh darn it, hold on. These are going to have smaller diameters. You know, that's kind of an anatomical thing, but that is related to why these contract more weakly. They're smaller in diameter, less myofibrils, less overall actin and myosin cross bridges pulling at the same time. The other big difference between them is how they make their ATP. In fast twitch, because you have lots more myofibrils going on, needing lots more ATP all at once, these are going to be using the anaerobic pathways to make their ATP. Whereas this uses cellular respiration. You know, that is going to lead to another big difference, functional difference between them. If you are using cellular respiration to make your ATP, you could just keep going and going and going and going. It's the classic way that your body makes ATP. So these have high endurance. If you're using these anaerobic pathways, we just talked about, after a minute and a half, you're in oxygen debt, you need to recover. So these are going to fatigue quickly. So even though they contract quickly and strong, you can't keep it up. Slow twitch contract more slowly, have weaker contractions, but they can keep going and going. You know, muscles that are for your posture, for instance, they're going to totally be these red fibers. The muscles that you're using for impulsive kind of things are kind of white. More things, I'm going to erase this to add a few more things here just because I'm running out of space. So I'm going to So if I am using aerobic pathways, if I'm using aerobic pathways in my slow twitch, what kind of organelles am I going to need to have to make that happen?
Mitochondria.
Totally, mitochondria. And you're going to have more, blood vessels coming in to supply the oxygen. There is a oxygen binding molecule called myoglobin in the cell that is what makes it red. It's why it's red fiber. Myoglobin is like hemoglobin, except it's localized in the muscle. So this binds oxygen in the tissue. So these are things that would be specific to slow twitch because they use aerobic pathways. For fast twitch, what are we going to have instead? They're going to have lots of creatine phosphate. AC. Right, so creatine phosphate, which is going to be used for the phosphagen system. What do you need to have a lot directly if you're going to be doing glycolysis?
Glucose.
Glucose. And what's the kind of way you store lots of glucose in big kind of branching chains?
Glycogen.
This is going to have lots of glycogen granules. Part of that whole carbo loading, when people do carbo loading, it's about making sure that your muscles are totally stocked up on all their little glycogen granule stores. So they have a lot to kind of pull from. So this is the two main kinds. If you want to think about this in terms of what you're familiar with, slow twitch. Think about slow twitch. This is basically dark meat. and white muscle. That's basically the white meat if you're looking at a chicken. You know, where do you find the dark meat on a chicken and the white meat on a chicken? Oh, somebody's got it.
The legs are, and then the thighs are dark meat.
Yeah, so I guess maybe in Marin, a lot of people are vegetarian and aren't as familiar with this. So if you're like having the poke, the chicken leg is dark meat. That's slow twitch. You know, that is how a chicken spends most of its time. It's just kind of walking around, scratching, trying to find some grain or some worms or something, bugs. So its legs need to have, you know, this constant kind of moving, walking, high endurance. They don't use their legs to sprint or anything like an ostrich or something or kick somebody. You know, as opposed to the white meat is the breast meat. That's doing the wings. If you've ever seen, how does a chicken use its wings? If you've ever seen Rocky, when Rocky's trying to catch a chicken, you know, what does a chicken do with its wings? Does it do like this?
They flap around like impulses, kind of, you know, just
Totally.
really fast.
This impulsive, like again, if you remember from when Mickey's having Rocky go into the chicken yard and try to catch a chicken, and as soon as he tries to catch the chicken, it kind of goes away, but it's not sustained. It's like this impulsive, powerful, but doesn't last very long. And then the chicken lands out of Rocky's reach, and then the chicken goes back and starts walking around again. If you think about other birds, though, has anybody ever eaten a duck?
Unfortunately,
So what does the breast meat look like in a duck?
it's super dark and gamey.
It's super dark. Exactly. Because if you're a duck, you actually, those muscles that run your wings, they gotta be endurance, because you're spending a lot of your time just like flying and kind of migrating and going from place to place. So you need to have dark meat for your breast meat if you're a duck. So you have endurance for your flying. Whereas if you're a chicken, you've got white meat for your breast meat, because your wings are about this impulsive but not sustainable, strong, like just get out of danger. So just if you're kind of thinking about it, you know, and most muscles, some muscles, particularly like postural muscles and stuff, tend to be predominantly red. Other muscles are more white. Most muscles have a combination of both fibers. What kind of distribution of dark meat, I should just say red fibers and white fibers, slow, fast twitch fibers you got is genetically determined. So, and different people have different distributions. So in what kind of activity you do is going to preferentially develop one or the other of what you've got. So if you're doing like really like low reps with high weights, that's going to be developing your fast twitch, this kind of, you know, powerful but not sustainable kind of muscle fibers. If you're doing low reps, high repetitions at low weights, then you're talking about, you know, slow twitch. But given whatever your genetic deal of the cards is, some people, no matter what kind of training they're going to do, are just going to be able to max out as a better distance runner or a better sprinter just based on their, you know, what they got in the genetic dealing in terms of their distribution of slow versus fast twitch fibers in their muscles. So, you know, so it's one of those, even though we talk about you can be anything you want, or you can, you know, just, you know, it's not total. You might, depending on what you kind of got dealt, you might end up being a better sprinter or a better distance runner than somebody else if you do the exact same training. So, any questions about muscles? Trying to think if, I'm going to go into my notes really quick and see if there's anything else that's critical, mission critical about muscles that we need to cover here.
Is our test going to be through this point?
Yes. So in fact, let me, good call. So it's going to be so I shouldn't just, it's not just CNS, it's going to be all of the nervous system. Plus what we've talked about from the muscles. Actually, oh, I take it back. I'm sorry. Test material does not end here because I didn't talk about smooth muscle. Test material, all this, plus smooth muscle that we're going to talk about in 2 seconds. But that's only going to be like another 5 or 10 minutes of material. So let me do smooth muscle right now. Smooth muscle cells are really different. And again, in the interest of time, I'm probably going to make this a little quicker than I might normally. It's still actin and myosin. But the actin and the myosin are assembled in really different kind of ways. So like in this picture I'm drawing here, actin is green. Myosin is going to be blue. The myosin is in between these little pockets of actin, but it's going in this more kind of lattice kind of structure. So when this thing contracts, it's going to more just squish in rather than elongate in a linear fashion. And also the way that you can stimulate this thing are lots of different ways compared to skeletal muscle. Skeletal muscle has one way to stimulate it, a somatic motor neuron. Smooth muscle, you can stimulate many different ways. So let's talk about ways to stimulate smooth muscle. And just for completeness, I am going to add in one more little word for you all. The little anchor points where all of the actin things are connecting, those are called dense bodies. So I like to think of them like the Z lines, because as the myosin pulls in against the actin, all the dense bodies get pulled closer together. So that's why if you're trying to think about, oh yeah, if the myosin is grabbing on the actin, all these little dense bodies are all getting pulled in, this whole cell is getting squished in. So their smooth muscle cell does not look striated or striped because you don't have that linear alternating arrangement of actin and myosin. If we look at smooth muscle cells, these are all my smooth muscle cells. They tend to have gap junctions connecting them. What does that mean? What does a gap junction do?
So electrical signals can be sent?
Yeah, so it's going to electrically connect. It means if this muscle cell here is depolarized, zzz, that is going to directly excite its neighbor. So in general, rather than having motor units like we were just talking about, most smooth muscle is what we call single unit, where you just basically, even if you are using a motor neuron, if this is an autonomic motor neuron, you have these things called diffuse synapses, where you just have this thing basically kind of puff neurotransmitter, whether it's acetylcholine or norepinephrine, kind of here. This is what we call a diffuse synapse. These little varicosities, these little swellings, are just releasing this neurotransmitter here. And exciting the whole family, and they're all co-exciting each other because they all have the gap junctions, so they're all electrically connected. So this is something that's pretty different. They are all electrically connected and they co-excite each other. And so one way to excite smooth muscle is through an autonomic motor neuron, but there are some other ways. So ways to excite smooth muscle. So one is autonomic motor neuron. But lots of other things can do it. Chemicals. We're going to see when we look at cardiovascular system soon that just levels of oxygen or carbon dioxide or things like that can excite or relax smooth muscle. Hormones. It's kind of chemical, right? Having epinephrine or having acetylcholine is going to, or I should say, having epinephrine is going to actually make your smooth muscle in your arteries constrict and things like that. So you can have things constrict just due to hormones. Stretch. This is actually going to be important when we look at autoregulation of blood flow when we get to the cardiovascular system. By stretch, I mean is if you have some smooth muscle here and you kind of stretch, you push on it. Let's say this is a wall made out of smooth muscle. And then I push on it and kind of stretch it out. It's going to respond by pushing back at me. So if this is going to be important in the blood vessels. Again, if you have a blood vessel and the blood is starting to push out and stretching the blood vessel wall, making it bigger, the blood vessel is going to automatically respond by actually pushing back and counteracting the force that's actually causing the muscle cells in the wall to stretch. So we'll come back and look at that. So chemicals, hormones, stretch. So just to kind of put that out there, smooth muscle has more than one way to get it excited. And the last thing I'll talk about with smooth muscle that makes it different than skeletal muscle. Smooth muscle can, I'm going to go to my typing because it's going to be faster. Smooth muscle can sustain contractions using only like 1% of the ATP needed by skeletal muscle. However, it contracts, 30 times more slowly. So this is going to be important because a lot of the things smooth muscle is doing, you want to just hold the diameter of your blood vessel. maybe adjust it a little narrow or a little wider, but it's doing a lot of stuff where it's just maintaining some particular contraction. You know, right now, your sphincters are squeezing shut to make sure you're not like peeing on yourself or whatever. And they just need to keep contracting until it's time to relax them. But there's a lot of smooth muscle that needs to sustain contractions, to hold the diameter of a tube or something. And there's this, again, it's a special way the myosin kind of latches on that's different than in skeletal muscle. It allows it to hold its contraction without having to keep burning a lot of ATP the way skeletal muscle would. You know, that being said, it does not contract as fast as skeletal muscle, but for what it needs to do, that's fine. So now I can say, test material.
So skeletal muscle has like one way to be excited, and then the smooth muscle has multiple.
Exactly. Skeletal muscle is only excited by a motor neuron, a somatic motor neuron. There's no other way to do it. Smooth muscle does get excited by motor neurons. At the same time, it's often excited by hormones or by just chemical conditions surrounding it, or by getting stretched even. So,
And then you said when they contract, actin, they're all coming closer together.
yeah, so if I go back to the that picture of, so it's still going to be actin and myosin, and there's still little myosin heads in here that are pulling on the actin and pulling the dense bodies closer together.
Okay.
But instead of like just getting shorter, it's going to get more kind of pudgy because it's getting pulled in many directions. So that is kind of the classic. Yeah, so it's still, and there's lots of other things, like rather than having a sarcoplasmic reticulum, you have these little things, the calveoli with like, you know, calcium's coming in from the outside, and there's other differences we're not going to go into. But at the core for smooth muscle, you should know still actin and myosin, but arranged in a different way. That's why it doesn't look striped. You should know that, you know, it gets activated in these kind of, usually in these big families. There are a few cases of what we call multi-unit smooth muscle, like controlling the diameter of your pupil with your iris and things, but it's not so common.
Is there also the release of calcium and like the troponin and tropomyosin?
So there's still calcium is important, but rather than coming like from the sarcoplasmic reticulum, it's a lot of times it's actually coming in from outside the cell.
Okay. Do we need to know those details like we
No,
did?
you do not need to know those details at all. So the only details you need to know for smooth muscle is what I've just said specifically right now.
Okay.
So let's take a break.